Okay, so welcome to the next in our guest speaker series. As a bit of background about Oxford Brain Diagnostics, we're a company spun out from Oxford University. We use a method for analyzing brain MRI that is based on changes in cellular structure. And we're working with pharma and biotechs and towards making available a tool for clinicians to help them correctly detect and classify Alzheimer's disease and other neurodegenerative diseases. But today we're delighted to have Professor Michael Weiner join us. Mike, I believe you started out at Johns Hopkins University in the 60s and studied and worked in medicine at Mount Sinai and Yale before transitioning from east to west coast of the States in the early 70s. And in '83, you established the magnetic resonance unit at the San Francisco VA Medical Center. And you're now professor in residence in radiology, biomedical imaging for medicine, psychiatry and neurology at University of California, San Francisco. So a long and distinguished career, and you're now a principal investigator, of course, of ADNI, and you were this year's recipient of the Lifetime Achievement Award at the Alzheimer's Association International Conference as well, which we attended, and that was great to see.
Summarizes that we have a group of very distinguished investigators who are measuring these brain volumes and put their measurements on the database. So Nick Fox at the University College London does something called boundary shift integral. Yutugu Tosin at the San Francisco VA has free surfer analysis. Paul Thompson at USC is using tensor-based morphometry, and the Mayo Group. Charles DeCarli is measuring cerebrovascular disease. The Mayo Group is looking at ARIA-H, that is with the microbleeds. Arterial spin label perfusion imaging is analyzed by Dr. Tosin at UCSF. Task-free MRI is measured at the Mayo Group, and diffusion by Paul Thompson's group. And hippocampal subfields by Paul Yuskowitz at the University of Pennsylvania. So each of these groups has put up literally thousands of measurements of each of these types of scans. But if you are a laboratory that's into imaging, you can download the raw data and you can use your own methods or any method you want to analyze the data. And this just makes the point that none of these protocols have been fixed. So for example, just to say, the FLAIR scan started with 2D and now they're all done with 3D. And with the arterial spin label, you know, just remember, we have Siemens scanners, we have GE scanners, and we have Philips scanners. So we have different, they're all a little bit different. Unfortunately, they are not highly standardized. And this is one of the problems with using this kind of data. Another issue which has just come up more recently is as the scans have gotten better, it's possible to reconstruct the face from the MRI scan, and you could match the face to somebody's photo. And there is a concern that because of this, people who are enrolled in ADNI, whose scans are freely available, could be identified and there will be a loss of privacy. So a number of groups have developed what are called defacing methods, where the face is removed from the scan. And we've decided to use a method that was developed at the Mayo Clinic by Chris Schwartz, which takes the patient's face off and puts a common face there instead. And we plan to implement that for ADNI 4. A problem with removing the face, though, is it can affect the way the actual image is processed. And so we've done quite a bit of work on this, and a paper is being written about it to show that the method that Dr. Schwartz is implementing is having a minimal impact on the actual brain data. And we plan to only release defaced scans for ADNI 4. Moving over to PET, I think that the single biggest contribution of the Alzheimer's disease neuroimaging initiative has really been to provide a large data set where people are amyloid phenotyped using amyloid PET and cerebrospinal fluid obtained by lumbar puncture to determine whether somebody is amyloid positive or amyloid negative, because people who are amyloid positive have, obviously, they have amyloid pathology. They have Alzheimer's disease, whereas people who have absence of amyloid in the brain do not. Now, it's a continuous thing, but we tend to use a cutoff that defines whether somebody is amyloid positive and amyloid negative. We provide our cutoffs, or you can use the data to determine your own cutoffs. We provide the continuous data for the amyloid PET. It's called SUVR data, in which the intensity in a particular brain region is referenced to the cerebellum. And we also provide the continuous measures of amyloid from the cerebrospinal fluid measurements that I'll talk about a little later. This shows the PET scans that are done in ADNI. So this is a huge repository of PET scans. We believe we've done the most PET scans of the brain of any project in the world in the history of PET. For example, for fluorodeoxyglucose, that is the labeled sugar that is used to look at brain activity, it's kind of a functional measure. There's 1,622 baseline scans, and you can see large numbers of longitudinal scans. Now, we have more than 145 scans out 6 years. So if you're interested in doing longitudinal FDG PET, there's a huge repository here. More than 3,600 scans, PET scans just alone. Florbetapir, which is the amyloid ligand that's produced by Avid Pharmaceuticals, owned by Lilly, we have 1,325 baseline scans and a large number of longitudinal scans. Not as many as FDG PET, because we only started doing this in ADNI 2. Florbetapen, which is the other amyloid imaging agent, we have fewer numbers, but a fair number if you want to do some kinds of comparisons. Fluortaucipir, which is the Avid Lilly tau ligand. Again, this is a very, very large repository of data, which is freely available to everybody. 837 baseline scans and a substantial number of longitudinal scans, over 1,200 scans total. So there are literally hundreds of papers now on all of this, and I can't even begin to summarize the results. But I'll tell you a few high points that I think are interesting. This shows you the amyloid status by clinical diagnosis. So this here is a scale showing the baseline florbetapir SUVR. This is the intensity scale with a very high amount of amyloid out at this end and a very low amount of amyloid at this end. And this dashed line here is the cutoff between amyloid positive and amyloid negative that has been determined by Avid Lilly and by our PET core. And so the first point to look at is, let's look at just normal people. These are normal, healthy people, and I'm probably in here somewhere, who are with the average age of 75 years. And what you see is the majority of the normal people have low amyloid, but there's a substantial number of people who are completely cognitively normal who have high amyloid levels, similar to the high amyloid levels that you would see in people with dementia due to Alzheimer's disease. So about 27% of the normal population is amyloid positive, age 75. Basically what that means is that these normal people who seem to be normal have Alzheimer's disease pathology growing in their brain. It just hasn't started to produce any symptoms or cognitive impairment. We call that preclinical Alzheimer's disease. The people with subjective memory complaints, it's a small number. They don't seem to have increased amounts of amyloid in their brains. We broke down mild cognitive impairment to early MCI and late MCI, just depending on the severity of the cognitive impairment, using a paragraph recall to measure memory cognition. In people with early MCI, only about 45% of them are positive with amyloid, and people with late MCI, about 60% are amyloid positive. But not clearly, not everybody with MCI, even though it's amnestic MCI, are amyloid positive. So you're having a lot of MCI that seems to be caused by other causes, not Alzheimer's pathology. And then when you look at the people with dementia due to Alzheimer's disease, in this case, 187, 85% of them were amyloid positive by Florbetapir, but 15% were negative. In other words, these people were diagnosed as having Alzheimer's disease clinically, but they didn't have Alzheimer's disease. So it's a false positive diagnosis. But this next slide shows the effects of the ApoE status of people. ApoE status has a big influence on amyloid positivity and probably is a major determinant of why ApoE is associated with more Alzheimer's dementia, okay? If you look at normal people who are ApoE4 positive, almost half of them are amyloid positive and half of them are amyloid negative. But if you take a normal person who's ApoE4 negative, only 21% of them are positive. So it's better not to be ApoE4 positive. Only 20% of the population is ApoE4 positive, but it greatly increases your risk at age 75 to be amyloid positive. Let's just jump down to dementia here and look at these same people with dementia. If people with dementia are ApoE positive, ApoE4 positive, 98% are Florbetapir positive. So if somebody has dementia and has got an ApoE4, there's only a 2% chance that they are amyloid negative. You almost don't need to do an amyloid scan on somebody like that. And remember, 66% or 60% of people with AD are ApoE4 positive. But if you have dementia and you're ApoE4 negative, now only 60% of these people are amyloid positive. So if somebody has dementia and they're ApoE4 negative, it's very hard to say that they have Alzheimer's disease, because 40% of people with dementia who are ApoE4 negative do not have Alzheimer's disease. Only 58% are positive. And the MCI population is kind of in between here. So even if you have advanced late MCI, if you're ApoE4 positive, now your chances are almost 90% that you've got Alzheimer's, but 40% don't have it, okay? If 40% of people who are late MCI who are ApoE negative, only 40% are amyloid positive, so 60% are amyloid negative. So this is a big contribution of ADNI showing this, and many papers written on this. This is a slide from Bill Jagis's lab using the longitudinal data we have on amyloid PET to show the time course of amyloid changes over the full course of the disease. And basically, this is showing the rate of amyloid accumulation over time. And this shows the absolute measurement of amyloid beta PET over time. And basically, the point is that as amyloid accumulates, the rate goes up, and then the rate of amyloid accumulation starts slowing. And the other point is that all of this happens many years before people develop mild cognitive impairment and dementia. We also have a lot of tau PET scans and longitudinal tau PET scans. One very interesting observation by the tau PET core run by Bill Jagis is that if you look at young people, average age of 39, who have Alzheimer's disease, they have very, very high tau PET. But if you look at people from Alzheimer's Disease Neuroimaging Initiative, average age 73, the amount of tau in the brain is much less compared to the young people. And many other people have shown this, that tau levels in older people with Alzheimer's disease are not so high. And that suggests that the dementia of older people with Alzheimer's disease is caused not just by amyloid and tau, but perhaps by other pathologies, including cerebral vascular disease and alpha-synuclein, or so-called Lewy body pathology, or something called TDP-43. I'm now going to transition and talk about the fluid biomarkers in ADNI, because ADNI is one of the very few studies that has MRI and amyloid PET and also fluid biomarkers, including cerebrospinal fluid and plasma. For cerebrospinal fluid, which is obtained by lumbar puncture, the biomarker core run by Les Shaw and John Cherzenowski has measured amyloid beta 40 and 42 and also shows you the 40/42 ratio. They've measured phospho-tau and total tau, phospho-tau 181, and they've used different assays and began with Luminex and then transitioned more recently to the Roche Alexis platform. So that's all of that information is in the ADNI database, and many participants had lumbar punctures and amyloid and tau PET scans, so you can look at the relationship of what you see with cerebrospinal fluid and what you see with the imaging markers. We also have frozen huge amounts of plasma, which is all aliquoted. And right now, there's data on amyloid beta 42/40 on some of the patients. Kai Bleno's lab has reported a large number of plasma p-tau 181, many hundreds of people, I believe many thousands of samples because he's got longitudinal data, and a large amount of neurofilament light data. This shows some results of the amyloid and tau in the cerebrospinal fluid. And the normal controls are down here at the bottom. And if you look at the amyloid level in the normal controls, you see it's 206. It drops to 164 in the MCI and drops to 143 in the dementia population, whereas tau levels go up. So it's interesting, as amyloid levels in the brain go up with amyloid PET, the amyloid levels in the CSF drop because it's thought that the A beta 42 is sticking to the plaques in the brain, whereas the tau levels go up as there's more tau. This slide basically makes the point that there's high concordance between the amyloid in the CSF, this is the amyloid 42 measured by Alexis, and the florbetapir concentration. So people who are amyloid positive, for example, up here by PET, are also going to be amyloid positive by CSF because CSF goes down. And people who are amyloid negative by PET, who are low, also are negative by CSF. There are a few people here in, sorry about that. There are a few people here who are outliers who are either positive with one or positive with the other. And there's some evidence, but it's debatable, as to whether or not the CSF detects the disease earlier, whether the CSF measurements of amyloid begin to come down before the amyloid PET shows positivity. There are some papers looking at that. This is some data from the biomarker core, basically making the point that the progression of the disease is very much affected by their biomarker status. That people who are amyloid and tau and neurodegeneration negative have relatively low progression, but that people who are amyloid and tau and neurodegeneration positive have much more rapid progression. So the more disease you have, the more rapidly you progress. Makes sense. Another point which many people are not aware of is that the ADNI samples, including the cerebrospinal fluid samples, the plasma, the serum, the whole blood, are available to investigators. Your laboratory, if you've got an assay that you'd like to run on ADNI samples, you can make a request to ADNI. It will be reviewed by a committee, and the National Institute of Aging is the final approval. I don't have anything to do with that decision. This is a list of all the different labs and all the different companies who have received samples from ADNI, and the results of all these various analyses are now on the ADNI database, because if you get the samples, you have to report the data back to ADNI. The samples are provided to you blindly, so in order for you to get the final results, you have to give the results back to ADNI, and then the results are posted. So I encourage people with new interesting assays. We are especially interested in assays for alpha-synuclein, which is the cause of Parkinson's disease, and the cause of Lewy body dementia. It's an important co-pathology in people with dementia, and but there are many very interesting measures of inflammation that could be studied. So we have a lot of samples, and we want people to use them. So what's going on with plasma? Because this is the real big exciting news in Alzheimer's disease. The fact that we can now you can get a lot of information as to whether somebody has Alzheimer's disease pathology just from a blood test. So the data is pretty convincing that plasma amyloid 42/40 is quite predictive of the brain level. And this has been done with ADNI samples and many other projects using either amyloid PET or cerebrospinal fluid as the gold standard. The amyloid 42/40 can be measured by mass spectroscopy. And in fact, in the United States, C2N is a company that does this, and the patient samples can be diagnosed, but there are also immunoassays. There is a sense right now that the mass spec assays are a little bit better than the immunoassays, but that could change. We don't think that this is dependent on the clinical stage. That is, if you have dementia or you are a completely normal person, the relationship of the plasma 42/40 to the brain level seems to be the same. So that's the plasma amyloid. Next is neurofilament light. Neurofilament light is associated with neurodegeneration and predicts neurodegeneration. It is not specific for Alzheimer's disease. Neurofilament light goes up in boxers, it goes up in multiple sclerosis, it goes up in stroke. So it's a neurodegeneration marker. It's not a specific Alzheimer's disease marker, but people with more advanced Alzheimer's disease with brain atrophy have higher neurofilament light levels. So it's an easy way to determine whether somebody has neurodegeneration. Phospho-tau 181 is a predictive of Alzheimer's disease pathology. And the newer assay called phospho-tau 217 seems to be perhaps even better. There's some debate as to which, and now there's another one, phospho-tau 231, I think it is, which is also predictive of so there's a lot of work going on, and we're not sure which of these is the best. Some of these, they may all be measuring different things. And one use of the ADNI samples is that we can do comparative measurements between different types of assays. I think that we all are coming around to understand that these blood tests, the amyloid, the neurofilament light, the tau, where basically we have A, T, and N here, just by blood, is going to be highly relevant for all kinds of aging studies, not just dementia studies, but for any study of an older person in psychiatry or people who are going to have surgery, people who have cancer. It's going to be very interesting to see how these markers of amyloid pathology and Alzheimer's disease in people are relevant to their disease progression. So this is going to become a huge area for all kinds of very interesting research. I just want to very quickly remind you that we have genetics. We have a genetics core. The ApoE is measured on every participant. A GWAS is measured on every participant. And whole genome sequencing is measured on much of the ADNI DNA. And there are very interesting correlations between various types of genes and various measures. And that's all in the ADNI database. Andy Sakin, the director of the genetics core, is very interested in a multi-omics update towards the system biology approach using the proteome and the metabolome and various other types of measurements, including the genome, and lots of different data has been obtained on ADNI participants. Finally, there's pathology, because that's really the gold standard as to whether or not people have a disease or not, is what is shown at autopsy and when the brain is harvested. Now, of course, we've had over 1,000 people in ADNI, but not that many have died, and we don't get autopsies on everybody. We right now have about, I think, 160 autopsies, but in the database right now there's 86 brains autopsied. And this shows you, so the beauty of this data is that you have autopsy data and then you have all the biomarker data. So it's possible to correlate the biomarker data with the autopsy data. And I should point out that not that many papers have been written on this very, very interesting data set. So this is a very interesting project. Just to make the point that of the 86 people we have autopsy, we're looking at their clinical diagnosis. 5 of those who died were normal, and 2 of those who died had MCI. 66 of the people who died with Alzheimer's disease, dementia, were thought clinically to have pure Alzheimer's dementia. But 10 of them, they were diagnosed clinically with dementia with Lewy bodies or DLB, Parkinson's disease, and one participant was clinically diagnosed as vascular dementia. One participant, interestingly, had progressive supranuclear palsy diagnosed in life, and 2 without clinical diagnosis. So this is the clinical diagnosis. Remember, just to make the point, the majority of the 86 people were diagnosed to have pure Alzheimer's disease. 10 of them were thought to have Alzheimer's disease with something else. But now we'll look at the correlations between the people who had Alzheimer's clinically and Alzheimer's disease measured pathologically. And there is a pretty good correlation between, for example, the people that were diagnosed as having pure Alzheimer's disease clinically, 53 of them had a high amount of Alzheimer's disease on autopsy. But there were 8 people who had low, or 1 person, none, Alzheimer's in their brain, even though they were diagnosed with Alzheimer's disease. So this shows you the data about the clinical diagnosis accuracy for making a pathological diagnosis. But one of the most interesting things is that if you look at the other diseases, we see that 36 cases had 31 cases, 36% of our cases had Lewy body disease, and 20% of our cases had vascular disease, and 10% of our cases had argillophilic grain disease, and 31% had TDP-43 inclusions. So there is a lot of co-pathology that is being detected pathologically that is not being detected clinically. And this points out a real weakness that we have in our field right now. We can measure amyloid, we can measure tau, we can measure neurodegeneration with MRI. We have no way to detect Lewy body disease or alpha-synuclein with a biomarker. We can't detect TDP-43 right now with a biomarker. We really need ways to do alpha-synuclein and TDP-43 by PET scanning or by blood testing. So that's a big need. And when people develop such tests, hopefully they can use ADNI data to validate them. So 86% of the population have additional pathologies, a very important point. Just to summarize the contributions of ADNI, we've provided a huge database of longitudinal clinical neuropsychological data, which enables clinical trial design and powering of trials. In my mind, clearly the biggest single contribution has been amyloid phenotyping using CSF, amyloid and amyloid PET, and tau phenotyping using tau PET. And I should add here, plasma, amyloid and plasma tau. We provide neurodegeneration phenotyping, structural, perfusion, diffusion, and functional MRI. We have helped improve the methodology and develop ways to do multi-site harmonization, which is very important for clinical trials. Our genetics data has provided imaging genetics correlations and the polygenic risk scores I didn't mention. We do pathology, and we have lots of plasma, and we have lots of plasma analyses. I think that the impact of ADNI is clearly due to the fact that we provide our data very without any embargo. It's very easy to get our data, and we encourage people to use it and write papers, resulting in 3,600 publications. We share our biofluid and genetic samples. ADNI is used often as a comparison data set to other studies. It's been used for PhD theses and career developments and over 3,600 publications. Furthermore, the whole ADNI model has launched a lot of other studies where people use the organizational structure of ADNI and some of the things that we do. So the Parkinson's Progressive Marker Initiative, or PPMI, funded by Michael J. Fox Foundation, was modeled on ADNI. A very famous project called the Dominantly Inherited Alzheimer's, the DIAN project. These are people with autosomal dominant Alzheimer's disease, run by John Morris and Randall Bateman, modeled on ADNI. Some projects with frontotemporal dementia, the multimodal imaging, a prediction of ADFT, and the so-called differential diagnosis PREDICT study is modeled on the ADNI approach. And more recently, a very interesting project called the Longitudinal Early Alzheimer's LEADS, directed by Liana Apostolodova and Gil Rabinovici, are modeled on ADNI. And we also are just concluding the Department of Defense ADNI, looking at the effects of traumatic brain injury and post-traumatic stress disorder on development of Alzheimer's disease in Vietnam veterans. These are 3 grants. All of the data is on the database. We're submitting our final paper, but basically the bottom line of this paper is that neither traumatic brain injury nor PTSD seem to be associated with more MCI. That they are associated with more MCI, but there was no change on AD biomarkers. We don't see an effect of traumatic brain injury or PTSD on Alzheimer's disease biomarkers. Well, what are the limitations of ADNI? The first is the generalizability. ADNI was designed as a clinical treatment trial, but many people were excluded because of comorbidities. So we excluded a lot of comorbidities, even though there still are a lot of comorbidities. Our population is primarily Caucasian and very well educated, average age, average education of 16 years. So we have a lot of people with master's degrees and PhDs. We have very few Blacks and Latinos. This does not really resemble the US population, and this is a big problem. So we need to change that. And on our next version of ADNI, we're taking great lengths, and we're going to have a very substantial budget to enroll more people of color, people, Blacks, Latinos, and people with low education. The other big limitation of this is that the cost of doing all these studies is very high. There's a big cost of recruiting people into clinical trials, and there's a high cost of the PET scans and the MRIs and lumbar punctures, et cetera. So ADNI is very expensive. Each ADNI grant has been $60 to $75 million over 5 years, and now the next ADNI grant is going to be even more. But in-clinic studies are very expensive. Recruitment costs are high. The cost of self-report, there's lots of self-report data gathered. A lot of neuropsychological testing is done. Why not use the internet to recruit and gather data from people at home? And this led to the creation of a separate project called the Brain Health Registry. I'm not going to talk about that more today, but I think especially with COVID, everybody's come to realize that remote testing and assessment of people at home using Zoom, using the phone, and using online assessment tools is scalable, it's effective, and it's going to become more and more important for clinical trials and also for routine diagnosis. I've already talked about this. The future of ADNI is to address the issue of low enrollment of Blacks, Latinos, Asians, and people with low education. Our next round will focus on recruitment of underrepresented populations. And we are going to use a lot of online recruitment using digital assessments. We're going to do a lot of plasma testing of amyloid 42/40, p-tau181, perhaps p-tau217, neurofilament light, possibly GFAP, to choose people who we think are the more likely to have Alzheimer's pathology, because the future of clinical trials is going to be using digital recruitment, online assessment, and blood tests to bring people into clinical trials. So in conclusion, since 2004, the Alzheimer's Disease Neuroimaging Initiative has longitudinally collected clinical, cognitive imaging, and fluid biomarker data, which has been widely shared, leading to over 3,600 publications. The ADNI data has been used to design clinical treatment trials. We encourage you to download.
So, Mike, following your presentation on the origins and outcomes so far of ADNI, we'll have a Q&A session. We'll be roughly at half an hour in the final cut, I think. And let's dive into some questions from the rest of the team here, if that's okay.
So before we get to Ian's question, I just want to say it's a pleasure to be talking to you, and I have a history of being in Oxford, which you don't know, but is buried there on my CV. Back in the late 60s, I got involved in biochemistry research, and I was trained as a nephrologist, so I was studying the biochemistry of the kidney. And then in the early 70s, I won some kind of an award that entitled me to spend 3 months in the laboratory of my choice anywhere in the world. So I decided I would work with the greatest biochemist in the world. And at that time, that was Sir Hans Krebs, who was still active at the Radcliffe Infirmary. And I contacted Professor Krebs, and he welcomed me. And I came to Oxford in the summer of 1975. There was a gentleman there who some of you may know by the name of Brian Ross. Does that name ring a bell to anybody? He worked with a guy named George Rada. You probably have heard of Rada, because Rada was really the pioneer in vivo NMR spectroscopy in Oxford. And he had a big program, big magnet and everything. And Brian Ross worked with him, but this was before that. This was before the NMR revolution started. So, and Brian had just moved out of his apartment in Parktown. I'm sure you all know where Parktown is. And so I moved into his apartment, and he had bought himself one of these thatch roof cottages up in Cassington. And I spent the most fabulous summer, the summer months there. I rented a bike. Oh, it was heaven. And of course, working with Krebs was amazing. And, I haven't been back to Oxford since. But I have many friends there who are still, unfortunately, I'm blocking the name of my friend who's an American who's there, who's an imager. Who went to work for GlaxoSmithKline and then came back to Oxford he's at the university. But I'm having a little age-associated memory name problem. Anyway, let's get to Ian raised his hand first. So Ian, go ahead.
Hi, Mike. Thanks so much for your time. You talked on your speech, obviously, about the difficulties of how many different MRI machines are used, and not just that, but how they have their head cores replaced or other changes, or they're upgraded at the same site. I was wondering if you collected the data on when the machines were ever turned off or when they ever had any kind of maintenance done, because while I understand that it's very difficult to get any formal information like that, even that kind of event is quite a big deal, when it comes to the MRI results. So I was wondering if you guys had anything like that.
First of all, the best way to get the answer to that question, well, the most direct way is just send me an email at michael.weiner@ucsf.edu, and I'll connect you with Clifford Jack, who's the head of the MRI core, and we can find out. I do not think that that's an interesting question. It's a good idea. But I do not think that we logged maintenance records I think that what is logged is what is on the header. The header information is really what we keep. So the header information does give you a lot of information about the pulse sequence. And I believe that the header hopefully gives information about the head coil. I think it does, but again, it's been a while since I was involved in those kinds of details. So the header certainly would not give you any information about the maintenance, and we don't, to my knowledge, all the information that we have in ADNI is what we would call patient-specific information. That is what relates to that particular patient, because that's the way the electronic data capture system is set up. So we don't have any way of recording information that is site general, but not patient specific. So something like a maintenance log, I don't know how that would be captured electronically in our system. And nobody ever thought about asking for that. So I'm sorry that we don't have that, but that's, in the early days of ADNI, we had the ADNI phantoms. You're familiar with that whole story that we did? We put a huge effort into this because of the issues of standardization and drift. So we worked with the people who at the time seemed to have a strong interest in creating head phantoms, and every site had to do some studies on phantoms. The phantoms were shipped all over the country, and for, I think in ADNI 1, there was even some adjustment of data to the phantom. But then some other studies were done to really evaluate the effectiveness, and of course the MRIs got better. And I believe that sometime as we got into ADNI 2, we stopped requiring a phantom scan we were requiring in ADNI 1 a phantom scan. Every time somebody went into the magnet, they had to have a phantom scan because there was that much concern about drift and changes and so forth. I think in ADNI 2, we just started asking for monthly phantom scans, and I don't even now know, I don't think we do that now at all because everything has gotten more stable. Again, some of the details are hazy on this to me, and Clifford Jack, who's at Mayo Clinic, who's, a very famous person in the field, and he is a radiologist. He really cares about all these MRI details. He could answer more detailed questions about that.
That's great. I appreciate it.
Thanks. Okay, who else has a question? Omar, go ahead.
Thank you, Stephen. Thank you, Mike, for your presentation. It was incredibly informative. I have two questions, but I'll probably ask the first one and then give some of my other colleagues an opportunity. You talked about diversity of population and cohorts in ADNI, which is pretty cool and pretty great at your sites. But I also noticed that ADNI is pretty widespread out throughout the world. Is there a plan for all the other sites to get equally diverse populations in their data sources, or do they just look to you and the folks in the US for guidance on that? How does that work?
Oh, okay. Well, this is a really good question. So let me a study of the entire population. It was intended to be a study of the kinds of people who get enrolled in clinical trials. And the kind of people that get enrolled in clinical trials are not the general population. Okay. I came to understand that at least in the United States, the people who volunteer to be in clinical trials are generally more affluent people. They're educated, they're affluent, and they have the time to participate in the clinical trial and the motivation. That fundamentally in the US means they're largely white, although we've always had some Black and Latino and Asian participants. But the average education of the ADNI participant is 16 years of education. That is 4 years of college, which means that half of the population has a master's, a PhD, or an MD degree. And that, if you look at the Blacks and the Latinos and the Asians in ADNI, you'll see that they have 4 years of college average. So I have said from day one that ADNI is not generalizable. It doesn't represent what's really going on. But my excuse was, well, this is the way clinical trials are, and the ADNI population looks like a clinical trial population. Well, things have changed over the last 20 years. And first of all, the politics have changed in the United States. There's much more concern about the issues of diversity because, in fact, some of these so-called underrepresented populations, especially the Blacks and Latinos, they're no longer minorities. In fact, in the state of California, where I'm sitting right now, I'm in the minority. Okay, Latinos are the major population in the state of California, and it's projected, I think, within 20 years that in fact Caucasians are going to be in the minority in the entire United States. this has got its own political ramifications, but that is the reality. But the second point is, which is what's most important, is that everybody in our field, including the people at the pharmaceutical companies, recognize that if we're going to make treatments that are going to be prescribed to the general population, that we need to do studies on the general population that are the people that receive the treatments. And it's just very obvious that poor people, and unfortunately in our country, because of our healthcare system and our economic system, we have a lot of poor people who are disproportionately Black and Latino, but there's plenty of poor white people as well. There's a big healthcare disparity problem. And poor people in America have more obesity, they have more hypertension, they have more strokes, they have more substance abuse, they just have more comorbidities. And those comorbidities clearly affect the way that the Alzheimer's disease pathology affects the brain. Because if you've been a healthy person like me, who was raised in a good environment with parents who cared about them, and I had good diets and a lot of education, and my hypertension's been always treated well, my cholesterol has always been kept low, etc. And I'm in my 80s now, okay, and I'm a really healthy 80-year-old, 81-year-old, okay. And so I know that my brain is very clean looking. I have no lacunes, strokes, and hardly any white matter disease. But you take your average 80-year-old ordinary Black person in America, and it's going to just be you're going to see a lot of cerebral vascular disease. So basically, the NIH is putting pressure, and the community is putting pressure, and everybody realizes we have to do more to be more diverse. So for ADNI 4, we got the message, and this is not easy work to do, but we were able to bring in some new leadership. Monica Rivera-Mint, Ozio Mancanquo. One is African, the other is a Latina. And we've done some work with what's called the Diversity Task Force. So we've increased our minority enrollment already in ADNI 3 to 21%, which is a big improvement from the 12% that it was just a few years ago. And we've decided to make this major commitment to enroll 50 to 60% of our new enrollees from the underrepresented populations. But we're not so foolish to say that we're going to be able to enroll people who match the demographics of the population in education and in income. Because I don't know what it's like in the United Kingdom, but in the US, if you look at the population over 60, 40% of the population in the USA over 60 has a high school education or less. A high school education or less. Very few people in ADNI have a high school education or less. And it's very difficult to enroll people with a high school education or less into the very biomarker intensive kinds of project that ADNI is, because people, they're living on very limited incomes. They just don't have the time, and they also are suspicious of the system and all these reasons. And the truth is, the only way it could really be done would be to pay people a lot of money. If you told people we'll give you $1,000 for every MRI scan, I think we could do it. But our ethics boards won't allow us to do that because it looks like coercion. That's a problem for another time. Now, in answer to Omar's question directly, there are these other international efforts, and I haven't seen any sign that they are moving in this direction. So we're just trying to set a good example. And it depends on the country. And for example, I haven't read anything about what's going on in the United Kingdom in this area. In France, they have a problem because the French culture is that everybody is French, and so you're not supposed to ask anybody what their religion is. So they don't even have data on what their diversity is, although they clearly have a large Muslim population there. And it's also very clear that their Muslim population doesn't have all the economic advantages in education that the Caucasian population has. Japan it's a secret, but Japan also has a problem, but they don't like to talk about it. They have a Korean population and some Chinese people. There are people there who are not given Japanese citizenship. There's disparities in every country. And but anyway, this is I think that the regulators are ultimately going to be the drivers of this, because if the regulators say, I'm not going to approve your treatment because the population that you ran your treatment trial on doesn't represent the population who's going to get the treatment, that's going to really get everybody's attention. And they are starting to move in that direction. So it's a good thing to be aware of. And it's a good thing to be aware of when you're planning your analyses, because we have to just recognize that older people with Alzheimer's disease have a lot more going on other than AD. They have synuclein pathology, which we have a lot of trouble diagnosing. That's a good challenge for your program, to try to figure out a way to diagnose who's got synuclein, who's got Lewy bodies. Because Lewy bodies are quite common in people with Alzheimer's disease. That's a very interesting issue that many people are not aware of. It was shown 10 years ago by a guy named Tom Montine in pathology that individuals who have genetic autosomal dominant early onset Alzheimer's disease from a APP or a mutation. So these people overexpress amyloid plaques because of their genetic abnormality. And they have plaques and tangles and neurodegeneration. They just get it in their 40s and 50s, and it's a rapidly progressive disease. Those people have a very high incidence of Lewy bodies. They have a lot of synuclein in their brain. And if you look at people with late onset Alzheimer's disease in their 70s and 80s, the non-genetic form, there's a very, very high incidence of Lewy body pathology. In fact, if you're into pathology studies, David Bennett demonstrated that people with dementia almost never have pure Alzheimer's disease pathology. People with dementia always have some other co-pathology. They'll have some significant cerebrovascular disease, or they'll have synuclein, or they'll have both. And then there's TDP-43, which produces a Alzheimer's-like, it produces hippocampal sclerosis and temporal parietal atrophy. It resembles Alzheimer's roughly in the MRI distribution, and it produces an amnestic syndrome. It's sometimes called late these days. And we currently have no way to diagnose the presence of Lewy bodies or the presence of TDP in people's brains. We don't have a PET scan ligand that is specific for those. And there have been some attempts. Duigu Tosin in our group has published some papers where she using pathology data sets or other information from, let's say, Parkinson's disease, has tried to identify certain MRI patterns which are associated with synuclein or associated with TDP-43, and then using that to try to infer the prevalence of synuclein or TDP-43 pathology in ADNI participants. I think she's published some of that. She's been using some AI approaches for that. I think that's a very fruitful area. But hopefully there will become blood tests or PET scans that are more specific for these pathologies. And of course, this whole issue of cerebrovascular disease and what's the best way to measure it, what's the best way to quantify strokes, lacunes, white matter lesions, using DTI to measure more subtle effects of cerebrovascular disease. This is really hot, especially because of the area which is associated with treatment. And this is probably right now the single most important thing that's going on in the field, because now there are reports. We heard a couple of weeks ago that somebody died from ARIA, from aducanumab, and now I just got an email yesterday that there's like 4 people who've died from ARIA. And the Alzheimer's Association is going to convene an emergency meeting to figure out, well, what do we do about this, and what kind of appropriate use guidelines do we put out? But this is probably what we expected, because when you do clinical trials, everything is very, very well controlled by protocol. Once you have a treatment that's approved by the regulators and is out there in the community, any doctor who knows nothing can prescribe it, and they can prescribe it in to the wrong people. And we need to be able to figure out who is more likely to get ARIA and perhaps who should be excluded from treatment. And so predicting ARIA, there's been very little published on this, and that's going to be a very, very interesting area. So I kind of wandered off of the topic of the question, but I think that this whole area of the diverse populations and the comorbidities, this is like aside from finding a treatment that works, and aside from finding better diagnostics, this is the most important topic in the field.
Thanks, Mike. Yeah.
I think Stephen's frozen. I had a question actually related specifically to cerebrovascular pathology, if I can jump in with that. So sure. One of the things that struck me from your talk actually was an interesting discrepancy within the subset who had pathology. So I think you said there were about 86 individuals within ADNI who'd gone on to have pathology. And in that subset, I think it was something like 20% had vascular pathology as well as AD. But from their clinical diagnosis, it was much less common. It was about 1 out of that 86 was clinically diagnosed with vascular pathology, and that just struck me as an interesting discrepancy. And I wasn't sure if that partly reflected kind of inadequacy of the procedures for detecting that pathology in life. Is it a limitation of what shows up on FLAIR? Is it perhaps even that the clinical diagnosis didn't use all of the available imaging? Is that something you could comment on?
Yes, I think that the clinical, well, first of all, the clinical diagnosis was done at baseline, and presumably the clinicians had access to the MRI that was done at the screen. But that's so variable. That's you have. We have 60 sites, we have all these different clinicians, we have some postdoctoral fellows who are looking at the scans, and what I don't know, Jed, is whether anybody has done a paper where they took those patients who had autopsies and who had cerebrovascular disease on autopsy, and then went back and looked at exactly what for example, Charlie DeCarli does the white matter lesion burden, okay? What was the white matter lesion burden in those participants? So in other words, we do have various scores. Paul Thompson does the DTI analyses. So his data should be there in the data set. I think there was certainly in ADNI 3, Clifford Jack's group started reading the microbleeds, and I believe perhaps giving some lacunar count information. This is all still done manually. Microbleeds and lacunar counts are just done by eyeball. That's an area where we certainly need something more automated. So I don't know. I don't know if anybody has actually done a kind of a careful well, I could say quantitative MRI to pathology correlation in that data set. But the truth is, Jed, that a huge amount of cerebrovascular disease is silent clinically. That people have a lot of white matter lesions, people have a lot of lacunar infarcts, and people can even have strokes, and it's never noticed clinically. So the only way that it would be detected for the clinical evaluation would be whether somebody looked at the MRI and looked carefully at the FLAIR and the T1 and did that right. And we don't have any control over that. So
cool.
Hope I answered your question.
Sounds like an interesting thing to look into in more detail. I have several other questions.
I should just mention that in addition to the 80 autopsies that are there now, I believe there's something like 140 or 150 participants that we have brains on that are being processed. So this is, and the pathology core is very busily working on this. And here's another little tip. We have authorized the purchase of a slide digitizer. So we plan to digitize all the slides of all the participants in ADNI, and all of that data will be on Loney as well. So there will be the opportunity for a lot of interesting analysis of that, especially the kinds of correlations which I described, looking at the MRI and the PET scans and how they predict or correlate with the pathology data.
If my signal doesn't cut out again, I was starting to say that your wide-ranging answer got onto neuropathology and comorbidities, which is where, I started out in neuropathology at the Radcliffe Infirmary, Mike, which you were reminiscing about earlier from your time in Oxford. I wondered, I hate to do this, but call people out, but Mario, did you have any questions about comorbidities? Because you've been looking at some neuropathology and comorbidities.
Yeah, absolutely. Thanks, Mike, for your very interesting talk. The neuropathological data for us, of course, represent the gold standard for a proper validation of novel AD biomarkers. And as you said, ADNI neuropathological data have been available in confirming AD, a very frequent overlapping co-pathologies. For the next round of ADNI, are you considering extending neuropathology to include additional information, for instance, about neuroinflammation or many column spacing, for instance?
I can't give you a detail on that, but once again, if you want to follow up on that, I already told you my email address. Just send me an email with that interest, and I will connect you with Richard Perrin, who is a professor of neuropathology at Washington University, works with John Morris. He is he's the new head of the neuropathology core, and he would be able to give you that. It's in the grant, the ADNI-4 grant, but I cannot. Normally, I would just give you a copy of the grant, but the grant has not been reviewed yet. So we are not going to distribute copies of the grant until we get the review completed, for obvious reasons. So, but we can get a quick answer to your question, and maybe you have some suggestions that he would add to the battery. So,
sure.
We, the neuropathology core does meet once a month. We have. It's interesting you bring this up. Maybe we should advertise to the world and open up the neuropathology core discussions to outside people like you. We do that in the other cores. The MRI core, do you know about that? Do you know that the MRI core meetings are attended by, I don't know, 50 people around the world? If you wanted to attend the MRI core, I don't know whether there'd be any objection. Well, in fact, if you're really interested in ADNI, if your company is interested in ADNI, you can join ADNI. This is a pitch now.
Yeah,
you can join ADNI. You can become part of the private partner scientific board. And I think if you join now, the charge is quite minimal. And starting in August, we're going to switch over, and the Alzheimer's Association will be convening the private partner scientific board, and anybody from any company can join. And I think it's like $8,000 a year or $10,000 a year, or maybe it's $10,000 over 5 years. I can't remember the fee, but it's low. And then you'd be a member of the whole ADNI, the whole industry group that is tied to ADNI. And so they have their own meetings. They have a lot of meetings. In fact, there is a group of people in industry just to represent the MRI people from all the companies, and like Bioclinica and Ixico, and of course the MRI people at Roche and Lilly, and you name it. So all those MRI people have their own little group, and they have their own meetings, and they have their own discussions, and they have their own work groups, and they make suggestions to us. And it would be a way to connect with people and learn about what's going on in this field. So But getting back to your question, we should think about opening up the pathology core to people. It just never occurred to me, and I'll do that. But I'm happy to get your question answered if you just email me.
Yeah, thank you. Thank you. Very kind.
That's great. Thanks, Mike. And I mean, I still have connections to the Oxford Brain Bank as well. And, you know, I think that would be quite nice if that was opened up as well. We could harness some expertise as well to contribute some thoughts to us as well. So I had a question that's been texted through, Mike. We have a colleague who's listening but not able to be visible. Terry, he's texted through a question, and he asks, does the current focus on liquid, and I think he's thinking particularly blood biomarkers, risk losing some of the benefits from imaging?
Well, we are not proposing to stop any of the imaging. We are proposing in ADNI 4 to do MRI every year, to do, I think, amyloid PET. I think it's every other year, or no, every third year, something like that. I mean, because the amyloid PET doesn't change that rapidly. And there's so much information that, you know, especially, for example, on cerebral vascular disease and other things. So, and in fact, if anything, the use of MRI is going to go up because of this ARIA issue. And this ARIA issue is not going to go away anytime soon. But amyloid PET, so right now there's a huge demand for amyloid PET for research purposes, and the clinical trials are putting a huge load on amyloid PET. This is what's rate limiting, at least in our country. And we have a lot of PET scanners. I'm sure in the UK and in other countries, it's even a bigger problem. Yeah. And tau PET is even more problematic. We've had a lot of problems just getting tracers shipped on time and so forth. So, so they're really in demand right now. But I wouldn't tell somebody who's a postdoc right now that you should make a long-term career in amyloid PET or tau PET. Because I think that the fluid biomarkers are gonna become so dominant. Now, though, I think amyloid PET is much more at risk to be replaced by the fluid biomarkers. I think the tau PET, at least in research, you get information on, just for those of you who are not into these PET scans, the amyloid PET is you get a big blob of amyloid. And the regional distribution of amyloid does not seem to be that, at least so far, to my knowledge, the regional distribution of amyloid doesn't seem to be very predictive or that informative. But the regional distribution of tau is critical, because tau highly correlates with neurodegeneration. So where you see tau, that's where you get neurodegeneration. And so tau PET gives you information that you can't possibly get from any a plasma measure. On the other hand, the question is, to what extent do you need the information in tau PET for diagnosis and treatment of patients? And I think right now you probably don't. Tau PET is being used in clinical trials mostly as an outcome measure, meaning that if you could see slowing of tau PET, that would mean that you have a treatment effect. And there was hopes in the beginning of tau PET that it was going to be a highly sensitive measure of treatment outcome, meaning that you could do a clinical trial with less participants and in a shorter period of time if you use tau PET compared with using clinical measures like a memory test. I think tau PET's been a little disappointing in that respect. It's not quite as robust or sensitive with low variance as we had hoped. Maybe that will get better, but so far it's been a bit of a disappointment. One big use of tau PET that Lilly recently did was, of course, Lilly owns fluortaucipir. They own Avid Pharmaceuticals. They used tau PET as an inclusion criteria in their phase 2 trial, which I thought was very clever. They only enrolled people who had a kind of a moderate amount of tau, not people with too much tau and not people with little tau, just people kind of with in between tau. And what that did was that allowed them to essentially have a population that was more homogeneous. There was less between subject variance. So they had more statistical power to measure their effects. And they were able to, of course, they have donanemab, which is a very, very powerful plaque-clearing antibody. And they were able, for the first time, to do a phase 2 trial, which actually met its primary endpoints. And in fact, in the history of Alzheimer's disease, clinical trials with disease-modifying treatments prior to donanemab, no phase 2 study ever met its primary endpoints. So that's part of the reason why we've had so much trouble having success. And the Lilly trial met it, but in part because they were very kind of selective of who they chose for that trial. But interestingly, they've not decided to use that same approach for phase 3. And it's pretty obvious why. You would not want to have on your label, you have to have a tau PET scan in order to get a drug. So when you set up any phase 3 trial, the inclusion and exclusion criteria you use in the phase 3 trial are ultimately good. The regulators are pretty much going to have you have that on the label. So you got to be careful about making it too difficult to get into the trial, because then nobody will really be able to get the treatment. So I think that there's, for academic people, there's going to be plenty to do with molecular imaging, and hopefully we will have scans for synuclein and TDP43, and hopefully we will have better scans for brain inflammation, and maybe even scans that can measure different kinds of brain inflammation and different, because inflammation in the brain is such a broad term. And as we all know, there's inflammation that's good, that clears misfolded proteins, and then there's inflammation that's bad, that's putting out cytokines, that's damaging neurons. And maybe these can be separated. It's not clear. So I think from an academic point of view, there's lots to do. I think, though, in a practical clinical environment, the fact that the fluid biomarkers are giving us so much information. I don't know if any of you were at the CTAD meeting in Boston just last month, but it just blew me away what's going on with fluid biomarkers. I have never been so excited. I went to my first scientific meeting in 1967. I've been to probably 300, 400 scientific meetings. I have never been so excited as I was at the CTAD meeting, because these fluid biomarkers are not just diagnostic, but they respond to plaque clearance, and then they also respond again when you stop the treatment. And they're giving a lot of interesting insights. And Susan Schindler at WashU had a really interesting talk in which she showed that the plasma markers and some longitudinal changes of plasma markers in normal, healthy people anticipated and predicted who was going to be amyloid positive. So that's just dynamite if that really holds up, because it will be a way to plan clinical trials to prevent development of symptoms, and hopefully even prevent accumulation of large amounts of amyloid and tau. So I think that anybody who's doing research who's not paying attention to fluid biomarkers has got their head in the sand. Yeah.
Mike, you've talked a lot there about amyloid and tau. A focus for us with our work on kind of regular MRI or DTI as well is to look at the neurodegeneration. So we're looking at that with imaging, of course, and I wondered, we're looking obviously at regional distribution and prediction of subsequent atrophy, because obviously atrophy tends to be after the horse has already bolted, right? We'd like to pick it up earlier, and that's what we think our sort of measure of quality rather than quantity of tissue does. It sort of detects the poorer quality that is likely to atrophy later. But I was wondering, in that context, what do you think of the sort of the subgrouping and the work that's been done to look at, maybe a continuum of multiple different types, which may have different distributions of not just tau, but other causes which result in neurodegeneration, just sort of varying across different subgroups?
Kind of research is worthy. It's I think that's important to do, but for me, but this is just my personal view, you just remember, I'm a kidney doctor. I'm trained in internal medicine. And what to me is really ultimately important is what can you use to really help the patient? And it comes down to diagnosis, choice of therapy, monitoring effects of therapy, predicting adverse effects, detecting adverse effects. And so then the question is, studies that are looking at heterogeneity and various subtypes of Alzheimer's disease and this and that, where does that take you? Where does that lead? How can what's the potential practical outcome of that? How could it be used in a clinical trial? Now, maybe you have ideas about that. I'm the author of many papers actually that have done this kind of thing. And maybe some of this heterogeneity, in fact, represents comorbidities that we don't know about. Maybe it represents genetic factors. So I think, I think there's a lot of what we call basic science work, lots of measurements and so forth. And then kind of out of that comes kind of practical uses. And if you're Oxford Brain Diagnostics is a company, so you've got something that you're trying to sell, right? You want creating value. So perhaps you suggesting this to your clients as something that that offers additional information for their trials.
Well, yeah, we certainly do that. But with regards to this question, I guess a couple of examples I was thinking of to sort of make the point might be, would perhaps one of those subgroups or subtypes will be more vulnerable to ARIA, or possibly, possibly it defines a window or a group, just like with the donanemab trial, where you might look at a sort of certain level of tau as one example, but you might also look at a certain distribution of alteration of the brain.
I agree. So you're probably talking to the companies and trying to get data sets and analyzing their data. And so, yeah, that makes a lot of sense. That makes total sense.
I'm envisaging a future where we have, more than just, one drug hitting amyloid. We probably have multiple of those, but also additional ones looking at, as you said, neuroinflammation or synaptic dysfunction, those kinds of things. So I'm imagining if there's a medley of drugs, you probably will increasingly want to refine how you apply them. That's what I guess.
Yes, I mean, fundamentally, that's what people call personalized medicine. It's been a jargon term. But rather, the fundamental idea is rather than just saying, well, somebody has Alzheimer's disease, they're in this group, so they're going to get treated like for Alzheimer's disease, period. We're going to try to more personalize it. Well, this has got Alzheimer's disease, but there's this type and they're vulnerable to this treatment, and therefore we want to avoid this treatment and give them that and so forth. I agree. I think doing that is a winning strategy. That's the future, in fact.
Yeah. Yeah, that's kind of the direction I'm thinking of.
And using all the genetic information, that's another good thing about ADNI is, we not only have the GWAS, but we have quite a bit of whole genome sequencing data. We also have some RNA sequencing data. I think that's in the database. Andy Sakin, our head of our genetics score, has been trying to get DNA methylation done. I don't know how important that is. But by the way, I just am looking at my clock and I realize that I have to be on another call in 2 minutes. So maybe if there's one more quick question, and if you have other questions, you guys are a lively audience. So if you have other questions, you can always email me. I'm happy. I'm very responsive on ADNI questions. Thanks,
Mike. I also had one eye on the clock as well. Does anyone have one question that we can give Mike like 60 seconds to answer? Maybe that's too much pressure.
Demetra, you're on mute.
Yes, hello, thank you. So yes, this topic has been discussed today, but the question I had in mind was about comorbidities. So in postmortem data, we see that many people had additional neuropathologies that were not clinically diagnosed. So do you think novel imaging measures could help to identify more of these pathologies in vivo?
Yes, and the answer is yes. And Tosin, I mentioned, is doing exactly that. I suggested to her a couple of years ago that she kind of pursue that. I think that's a very, very fruitful approach to try to inference, use as much information as you can. In fact, use the genetic information and the plasma information and the clinical information and the imaging information to predict who's got synuclein or TDP. For you guys are a terrific audience, and I enjoyed talking with you and wish you all a very happy weekend and happy holidays.